you so much for the introduction, Megan, Eric. Thank you for the invitation. I'm very passionate about biosensors. I think I found my people today. As Megan said, today we are going to talk about custom-made wholesale biosensors and a little bit about kill switches and reporting via bioelectrical devices. Before I start, I want to very briefly disclose a couple of conflicts of interest. I'm part of the intellectual property that was foundational for the creation of the company Tenza Bio and the startup from my postdoc lab. In the future, I hope I will receive royalties from that, and I want to disclose that. And also, I was the founder and CSO of a company that closed recently, but it's in the process of being terminated. It's a company that focuses on diagnostics of iron deficiency and treatment for young women athletes. Again, full disclosure there. Now, when my students ask me what I do in the lab, I tell them applied synthetic microbiology, and they are a bit puzzled. But if I have to summarize what we do, I call it sense and contain. We create biocontain wholesale biosensors that are based on regulators and microbial electrode interfaces. Our end goal is the creation of portable and wearable sensing devices that host cartridges of living bacteria that are pre-programmed to detect different biomarkers of interest and report the presence of said biomarkers by producing an electrical current. Now, I want to give you a little bit of background so that you understand what's my point of view of biosensors and how I arrived to the field. So as Megan said in the introduction, I'm an environmental microbiologist by training. So I spent a few years characterizing new catabolic pathways and also the specific and overimposed regulatory networks that would condition these pathways and when they would be expressed. So essentially, the part that interested me the most about the job was to figure out how cells would sense these aromatic compounds that we were degrading, to what concentrations, what was the threshold, and how that would inform the different clusters that would be expressed in the bacteria. For my postdoctoral work, I wanted to keep in the regulatory, working on the regulation side, but I wanted to leverage the tools of synthetic biology. Now, the particular flavor of biosensors that I work with are wholesale biosensors. As you know, wholesale biosensors possess all the modules that you can expect from any sensor for detection of the signal, processing of that signal, and then an actuator. And in wholesale biosensors, we can leverage the synthetic biology toolbox to improve all of these different modules. We can improve detection by working with receptors via protein selection, engineering of transporters and transcription factors. Regarding signal processing, excuse me, we can create genetic circuits as complicated as we want, essentially, to process this information and integrate it with the overall physiological state of the cell or any other signal of interest. And finally, we know that we can use different genes as reporters. Very often, we focus on luminescence, expression of fluorescent proteins, but we can be creative and work with changes in motility, release of different substrates, and of course, bioelectricity. Now, I was very interested in wholesale biosensors based on transcription factors and riboswitches because they bridge the detection and integration phase, so that with the same module, you are doing the detection and starting the processing of the signal. As you know, there are many, many beautiful examples of the possibilities that wholesale biosensors have in literature. I'm not going to enumerate them, but they are extremely creative. And essentially, there's a huge panoply of possibilities of things that we can do. I'm particularly interested, even though that's not the topic of this talk, on the combination of diagnostics via wholesale biosensors and the production of a molecule that is pharmacologically active and can treat whatever condition. But again, that would be a subject of discussion for a different conversation. Now, what brought me to work with biosensors initially in my postdoc was to answer the question of how can we use whole cell biosensors to detect molecules that are not recognized by any regulator that we know of. And of course, we always look to nature for inspiration, and from a very basic level, nature detects small soluble molecules, that's the ones I was interested in detecting, both in the, let's say, protein wall and the RNA wall. Of course, we have in the protein wall antibodies and transcription factors, and in the RNA wall, we have aptamers, secondary structures of RNA. So starting with transcription factors, of course, when we want to detect a new compound of interest, we can use transcriptional factors associated to clusters of synthesis or degradation of said compounds that are present in nature, and that's a fantastic option. But sometimes those clusters are unknown, or we don't know of any regulator that would recognize the compound. So I set my objective on during my postdoc on generating chimeric proteins, essentially new fusion genes in which I would take domains from transcription factors and domains from proteins that were not transcription factors, but would give me the ability to specifically recognize my small molecules of interest. And that's how I started to work with chimeras, which essentially are the symbol of my lab. As you know, the mythological chimera, as the genetic chimeras, is the amalgamation of parts from different animals. The mythological chimera, of course, a lion, a snake, and then a goat's head. But honestly, the goat's head grossed me out a little bit, so I let that out of the symbol of my lab. My chimeras and the apparition of fusion genes is one of the main tools that evolution have to create new activities, and there's enormous literature speaking about it, starting in the 70s, on how the creation of fusion genes gave place to some of the proteins, to many different families of proteins. So again, when I started in my postdoc, I said, okay, I want to develop a pipeline so that any lab in the world that is interested on constructing their own custom-made transcription factor can do it. And I started with three premises: simplicity, affordability, and high throughput that would increase our chances of success. Simplicity was twofold. I wanted the pipeline to be a straightforward process, and also simplicity came in the form of working with the most simple transcription factors that I knew of, which were monogenic transcriptional repressors. Price in this case consisted, or affordability consisted, on using the cheapest DNA synthesis that was available to me at the moment, which was synthesis of DNA in microarrays, the cheapest cost per base. And in terms of high throughput, it got translated into different steps of flow cytometry enrichment. So with that, I created a pipeline in which you would identify DNA binding domains from known transcriptional factors that had known operator sequences in promoters. And then you would identify ligand binding domains, which would be proteins not part of transcription factors that had been published to recognize your molecule of interest, or which could have the potential to recognize your molecule of interest. Then you would create a library of fusion genes, creating every possible combination of these DNA binding domains and ligand binding domains, connecting them via different linkers. In that particular publication, we constructed around 150,000 chimeric transcription factors in a single cloning event to create a huge library. In parallel, we were building a specific reporter plasmids for every DNA binding domain, and we brought it together into the same bacteria. We did a first negative enrichment to figure out which ones of our transcriptional repressors were able to repress the expression of our reporter gene. And then we expose the cells that we had recovered to the molecule of interest that we wanted to detect. In this case, still carrying over from my environmental microbiologist days, I wanted to detect benzoate, which is again an aromatic compound. In this case, we wanted to enrich for cells that would be able to de-repress, and we could iterate this process a few times. Finally, getting library after the enrichment that we would sequence and compare with the initial library to validate individual candidates. This process worked, and after years of development, we can say that it worked, and we propose two new transcriptional factors that would recognize benzoate, resulting of the fusion of the genes that you can see here, which was very fun, was important for me, demonstrated feasibility at the scale, but it had a few inconveniences. And it was that for a lab that was not a synthetic biology lab, the pipeline was still complicated. And for many humble labs or labs of modest means, it was still a fairly expensive process. So funny enough, normally you start in the smallest scale and you go to the biggest scale, but in this case, I went the other way around. I said, okay, how can I simplify this process? And what's the minimum amount that I can do to ensure some levels of success at the smallest possible scale so that people can actually use this approach. And hopefully we can learn from the design rules that we got from our initial publication. And then what would we learn with this approach? So at that point, that's when I started to become an independent scientist at Forsyth and at St. John's. I moved from big scale chimeragenesis to, let's say, affordable chimeragenesis or modest chimeragenesis, if you may. Now, at that point, my interests had shifted a little bit, and I wanted to expand the range of molecules that I wanted to detect. I have been working in environmental microbiology for a while, and now I wanted to detect biomarkers in saliva. The Forsyth Institute, where I was working at the time, it's a dental research institution. So there are a lot of experts there in oral microbiome, salivary diagnostics, and so on. So my biomarker needed to still be small soluble molecules. I wanted to focus on that. Of course, I'm very interested in detecting proteins, but that's a totally different fight. And at the time, many of my friends at Forsyth Institute and at different dental research institutions pointed to me that there was a need for early detection of oral squamous cell carcinoma, which is one of the most prevalent oral cancers. And actually, years later, I participated in an NSF I-Corps initiative in which I could validate with not with the research community, which I knew there was a need for the detection of this disease, but with actual oral surgeons and dentists, that there was a deep interest in detecting this condition. And also due to the nature of the biomarkers that have been proposed for the condition, detecting it via regulator-based whole cell biosensors seemed very, very feasible. So at that point, after consulting the literature, I decided to focus on the detection of N-acetylnuraminic acid. Its abundance in saliva is correlated with dysbiosis and oral squamous cell carcinoma. It's a very interesting molecule because our body secretes it, and many members of the oral microbiome engage in a behavior called sialic acid harvesting or scavenging, in which they are able to clip sialic acids from our cells and attach them to their cell envelope so that they can fly stealth under our immune system. So its overabundance in saliva has been correlated in many publications with tissue inflammation, oral cancer, and serious dysbiosis conducting to oral cancer as well. So as I said, I wanted to make the process affordable and bare bones. So I took only one DNA binding domain, the DNA binding domain from LACAI, as you well know, maybe the best studied transcriptional factor. And then I look in the literature for two proteins that were able to bind to neuraminic acid. None of them are transcription factors. These are soluble proteins associated to an ABC transporter and to a trap transporter. They are SAT-A and SIAP, and I connected them via in 5 different ways, 4 linkers of different biophysical properties, plus a direct connection between the domains. So as you can see, very bare bones approach, very focused on the particular domains that I was working on, rather than brute force my way through the project. So at that point, I created the library. I would love to say that I created the library with all the possibilities, but it turned out that certain combinations of LACI, linker, and these domains resulted extremely toxic for the cell, and we couldn't work with those combinations. And here I present to you a model 3D structure of one of them, one of these chimeras that my undergrads took the initiative of naming them with mythological creatures, many of them. So this is called Sphinx, which is a chimera of LACI, a link, relatively short and stiff linker, and then SIAP. Now, to test these fusion transcription factors, I use a very simple genetic circuit. I use a low copy plasmid that would express the chimera under the control of TET-R, which meant that I could open or close the expression of the chimera by adding ATC to the culture media, and the chimera could repress the expression of a fluorescent protein in a high copy plasmid. Essentially, this would be, if we represent over time relative fluorescence, this is fluorescence corrected by the growth of the culture. We would always compare these chimeras in 4 different situations. One is a basal state in which there is no anhydrotetracycline, nor there is inducer. So theta represents the chimera and GFP can be produced. So we have a basal signal. Then situation in which the chimera, if it's a functional repressor, would be allowed to repress. So we add ATC that enables the expression of the chimera. And if the chimera is functional, we observe a decrease in the GFP production. And finally, a de-repression stage in which we add neuraminic acid on top of anhydrotetracycline to the culture media, so that if the chimera is able to de-repress, this is sense neuraminic acid, we will recover part of our relative fluorescence signal. We perform experiments, as I said, with all of these chimeras, but I will focus on our best candidate, which was Sphinx. And both in vivo and in vitro data suggested that there was a specific interaction between Sphinx and neuraminic acid. We express it in E. coli. We had a good signal, and our preliminary in vitro data pointed to the same direction. And also, we studied the range of neuraminic acid that it could detect, and we studied between 0.5 millimolar to 5 millimolar, and we found a statistical significance in the reporting at 1 and 2 millimolar. This is interesting. Pathological concentrations of neuraminic acid are around 1 millimolar. Under 1 millimolar, they are not considered pathological. And I want to bring your attention, if any of you is interested on creating your custom-made transcription factor, creating chimeras, the dynamic range of these transcription factors out of the box is never going to be impressive, at least in my hands. Take into account that we are recapitulating the stage of evolution in which a new gene has appeared essentially by fusion, and it has not been subject to natural selection to refine the communication between the DNA binding domain and the ligand binding domain. So we were happy that we could see the signal, but we are also aware of the limitations of this type of technology. We also tested the specificity of our strain expressing Sphinx to detect different analog compounds besides neuraminic acid. The most common analog compounds that you can find in saliva are N-acetyl-manosamine and N-acetyl-glucosamine, and our biosensor didn't detect them. However, it gave a very strong signal for N-glycolyl neuraminic acid, which is very interesting. N-glycolineuraminic acid is very similar to an acetylnuraminic acid, has also been postulated as a biomarker of oral cancer. I just didn't focus on it in the beginning because N-glycolineuraminic acid we accumulate in saliva from the diet under certain circumstances. So it's not a molecule that our body naturally produces. It's more of a reaction to certain diseases and certain diets. So it's more complex as a biomarker. We also did some experiments with artificial saliva in which we mix artificial saliva preparation. This is very complex artificial saliva that contains most of the trace elements and macromolecules that you can find in saliva, including stabilized mucins. So it's a viscose, it's thick, it's as good as of a model of saliva as you can get. And we also get a very specific signal for around 1 and 2 millimolar. So we were really happy about that. In the future, we want to work with our collaborators at Rutgers University, Mizu and Carla Cugini, to actually test our strain detecting neuraminic acid in samples from patients. Now, a brief comment. We did some docking experiments to see if we could explain the binding of N-glycolineuraminic acid and we observed that simulations in the binding pocket of the chimera favor a stable binding of both neuraminic acid, whereas it didn't show a stable binding of N-acetylmanosamine and acetylglucosamine. So we could have essentially predicted the behavior that we were observing experimentally. Now, one of the problems of using biosensors for this kind of detection analysis is that is very slow, and that's something that we are always in conversation with referees and with founding agencies. So we are working on how to make the assays with hosted biosensors faster by using cells that have been pre-grown and trying to do assays that are as independent from bacterial growth as possible. I don't have a definitive answer, but with the last approaches that we've been doing, we've been able to shorten the time frame from 12 hours to 4 hours, which is still, if you ask me, a very, very long amount of time. But this is something that, of course, can be refined and we are working towards in the future. The other way that we are trying to shorten the detection time is by using bioelectricity, as I will explain in a couple of slides. We are currently collaborating with Professors Anne Vázquez and Frank Vázquez from the chemistry department here at St. John's on comparing functional chimeras that we constructed like Sphinx with chimeras like this one called Kunst that essentially have the same DNA binding domain, the same ligand binding domain. They only differ in the linker, and yet Kunst is totally incapable of detecting neuraminic acid. These are AlphaFold simulations, but we are working towards corroborating our initial observations by crystallography. Our current impression is that the linker in Kunst is very loose and it cannot stabilize properly the two domains of the protein in the proper 3D configuration, whereas in a Sphinx, there's some stabilizing salt bridges that enable the protein to latch properly into the operator and detach when detecting the neuraminic acid. This is very exciting and work in progress, truly. Now, I mentioned that in the lab, we are working with transition factors, and this neuraminic acid sensor has been our object of work for a few years now. I just wanted to tell you that also in our lab, we are currently working with detection of signals in the RNA world with aptamers. One of my graduate students is actually creating whole cell biosensors for another 2-3 salivary biomarkers based, in this case, in custom-made riboswitches rather than transcription factors. And the other way that we use riboswitches is for our biocontention aspect that I mentioned in the title of my talk via the incorporation of RNA thermometers to our circuit. We are very interested in making our whole cell biosensors biocontain. We want to limit this in case that we can assemble our sensing devices with our cartridges with living bacteria. Of course, we want to limit dispersal of the sensing bacteria after release, for user, operator, patient safety, for environmental safety, as well as for IP protection as well. So what we are doing is that we are incorporating RNA thermometers in kill switches that trigger the death of our biosensing strains if they are not stabilized at certain temperatures for a long period of time. That's the way that in this proof of concept, we are sensing that the conditions where we want them to be confined have changed. For that, we are using a very simple reliable switch arrangement in which when transcription happens with this RNA thermometer in the 5' untranslated region of toxic protein at non-permissive temperatures, the RNA thermometer acquires a secondary structure that prevents translation, whereas at permissive temperatures, it enables translation. And well, not of the toxin, sorry, but of an antitoxin. That's how we control survival of the cells. Interestingly, when we started to assemble these RNA thermometers, we work with wild-type thermometers, such as these mini rows that you see here. Now, wild-type RNA thermometers are fascinating pieces of regulation. One of the main sources we have of them are human pathogens, since one of the ways that human pathogens detect that they are inside of the host, this is us, is by a stable temperature at around 37 Celsius. Now, cloning these thermometers, working with them, it's a bit complicated. As you can observe, these strong secondary structures are a pain in terms of constructing circuits and preventing the apparition of mutations. And also, I suspect that the stability of these systems goes beyond the temperature required to melt them. So I think they also have other regulation implications, most of them that we don't fully understand. So my other graduate student has worked on the construction of new synthetic RNA thermometers that are easier to clone, more simple in their structure, and that are resulting very interesting to work with for these kill switches. We are working on the characterization of a collection of around 12, 14 synthetic RNA thermometers. We compare the activity at 37 Celsius that you can see here in red, and then at 23 Celsius as a proxy for kind of room temperature that you can see here in blue. We measure the time that the system is essentially closed at the low temperature, and we are in the ways of plotting which ones are the best in this library that we are creating. In this graph, you can see the delta of temperature in the x-axis and the logarithm of the relative fluorescence at 37 Celsius over 23. In green, you have an unregulated control that gives a signal. So everything to the right of that is temperature regulated, and everything closer to the x-axis keeps its strength at 37 Celsius once the system is open, which is essentially what we want. Now, just to wrap up, I mentioned, okay, we construct custom-made whole cell biosensors. We incorporate these biocontainment ribo switches based on temperature, but our final goal is to incorporate these sensing bacteria into devices in which they report the presence of the biomarkers via the generation of electricity. Exploiting the electrogenic ability of bacteria, me being an environmental microbiologist by training, has always been very interesting. Excuse me. And that's something that we are trying to optimize right now. There's many beautiful examples of publications by many groups in which you have bioelectrical interfaces for detection of different compounds. My particular objective is to substitute as many of the electronic modules by biological modules so that we can do detection and processing of the signal inside of the cell via genetic circuits. The cell would act as will be integrated in microbial fuel cells, so it will produce its own electricity that will feed the device and also at the same time serve as a reporter of the presence of the biomarker. This is very eagle's eye view proof of concept. But this is exactly what we want to achieve in the future. So we have different cartridges containing different lyophilized bacteria that are whole cell biosensors for different compounds. Now, the way that we are working with it is that we are playing with catabolic repression pathways to maximize NADH production and thus electricity generation in E. coli in response to the presence of neuraminic acid. The way E. coli generates electricity, by the way, is not like Geobacter or Shigella, where you have nanowires. The easiest way to generate electricity with E. coli is via electronophores. So essentially, you supercharge the bacteria with reducing power. Part of that reducing power is transferred to a soluble molecule that is able to leave the cytoplasm of the bacteria and donate those electrons to an electrode. And that's what we are playing with and what we are more interested in. Essentially, we are subordinating the catabolic repression pathways of E. coli to a new superstructure of overimposed regulation that essentially starts at the cusp with the transcription factor that would detect neuraminic acid or any other biomarker of interest. And we are at the stage in which we are starting to play with new toys, creating these small microbial fuel cells in the lab and testing our initial designs, which is extremely fun, if I might say it. And with that, I don't want to go over time. I want to thank you all very much for your attention. I want to thank my students who are the ones who have actually done the work, as well as my mentors and collaborators, Chris Johnstone from MD Anderson Cancer Center, Catherine Lemon from Baylor, and my colleagues at St. John's and Rutgers. And of course, I want to thank particularly Professor Lovely, who in some conversations many years ago guided me through the possibilities of bioelectricity as a reporter. So thank you so much, and I will take any questions that you may want to make. Thank you.